Hello, and welcome to the inaugural episode of CharaBio's Listen to the Biotech Expert series, where we bring you deep insights into the world of biotechnology to inspire and connect scientists, medical doctors, and biotech founders. I'm Fabio D'Agostino, founder and CEO of CharaBio, and your host for today, and we are thrilled to have you join us for this Virtual Fireside Chat. I'm excited to introduce our esteemed guest, for episode 1, Dr. Francesco Marincola, Chief Scientific Officer at Sonata Therapeutics and a true expert in oncology, immunology, and cell therapy. So, Franco, thank you for taking this interview today. And can you tell us about a bit about your journey so far?
Yes, well, thank you, Fabio, for inviting me, and thanks for asking for my view of the field. I'm old, so I've seen a lot of things in my life, so I'm not necessarily going to be right, but definitely there's a lot of experience. As they say, wisdom comes from experience comes from lack of wisdom. So, I think I have a lot of wisdom. That's the way.
Yeah.
But so I actually, after finishing medical school in Milan, I went to Stanford to do transplant immunology. That was my original interest. Heart transplant. Stanford, very famous for a heart transplant at that time with Norma Shumway. But as I was there and then doing my training, Steve Rosenberg became famous. I became more interested in tumor immunology. And the reason is because tumor immunology is more natural. It's a natural phenomenon. Like transplant immunologists, the body react to something and it's not there. It's kind of different interest. And plus cancer immunology has the cancer on the other side, which is very interesting from the biological standpoint. It's a very fast evolutionary process where you can understand all genetics at a high speed. So I became more interested, and so I decided during the training there at Stanford to spend some time at the NCI with Steve Rosenberg. And that was just to see, is it really true what people are talking about? And I was impressed because at that time, people are treated with interleukin-2, systemic interleukin-2, or maybe tumor infiltrating lymphocytes. But it was striking because maybe the responses were not that common, but they're common enough to be more than anecdote. So 1 out of 10 people actually responded in a long-term complete regression of the advanced metastatic melanoma, and that was striking to me. It was just to see something like this was so interesting. And although people were saying, well, it's rare, it's not a very broad effects in the larger population, and still thought, well, still interesting stuff to study, understand really what happens, why some people respond and some didn't. And so all my life I dedicated to try to understand, particularly in humans, more than experimental animal model. So focusing more on human genetics, immunogenetics. What's the difference? Why some people respond or not? And that's what my life pretty much. I spent most of my career at the NCI and then the NIH in general. I was tenured there, and I stayed there until eventually I reached retirement age, government retirement age. So I decided to do something else. We can talk about that later. But as I was there, a lot of things happened. So yeah, I witnessed the I was there actually. I participated in discovering a tumor-associated antigen, melanoma, EP100, and then all the vaccine studies that we did, understanding better what happens when people are vaccinated for specific antigens. And a little bit at a time, we understood the mechanism of rejection. So it was really interesting what happens when people reject their cancer. And turned out that, and we published that in 2008, we summarized the immunologic constant rejection, describing the mechanism, which is basically an autoimmune phenomenon against cancer. And it's just identical. If you look at the genes that are activated, there are about 500 genes. They're just the same you see in graft versus host disease, allograft rejection, acute flares of autoimmunity, clearance of pathogens, acute clearance of pathogens, for example, during acute hepatitis. Just the same. It's exactly a very natural, highly conserved mechanism across any species, across any treatment. Where you have rejection, that's what you need, are the requirements. So that was really interesting. At the same time, we discovered by doing biopsies before treatment, doing small findings without removing the tumor, we could follow what happened. So we were interested. It's different if you do biopsy before treatment, again, IL-2 TLs, to infiltrate lymphocytes. We can predict? And so the first paper that predicted, in fact, it showed that you can predict responsiveness was actually a paper we published in 2002, where we suggested that Anna Wang in my group was the first author, suggested that people that responded to immunotherapy had cancer that were already prepared to this responsiveness. They're already inflamed. And that's a concept now is accepted everywhere. And so the next step is being try to understand so what's special why some people have inflamed tumors to respond to me. They're more likely, not necessarily do, but more likely to respond to the cold. Tumors that are not inflamed almost never respond to immunotherapy. And so that's what the last few years of my career were about, understanding what started the mechanism, which in the end we summarized and we kind of affectionately calling the 4 Ps. You need 4 requirements. You need to have presence of T cells. If there are not T cells in the cancer, they're not going to go away. They have to penetrate the cancer. As you may know, there is a lot of cancer, a lot of T cells around, but not inside the cancer. If they don't get in the cancer, even Geronga and other groups have shown that there is no benefit of having T cells around them. The survival of the patient is not better. Likelihood of response is not any better. It's like having cold tumors. They also have to persist. They have to, T cells don't last long. That's part of it. It's natural nature of T cells that you want to persist forever. It can keep proliferating. So otherwise you have autoimmune disorders every time you have. That's them, and so they kind of, for a while, they expand and they go to a contraction phase. That's not. We have to do better than that. We have to intervene to make it last longer, many times. But most important, they have to be preponderance over immune suppression. So these are the 4 requirements, and only when all these 4 happen at the same time, you're going to see a complete response. So that's pretty much, if you know my intellectual story. And that's what we're interested in now. How can we basically follow what I call the blueprint of nature? How can we just repeat what happens as a natural phenomenon, make it happen more often by turning cold tumor or immune-excluded tumor into immune-infiltrated tumor? And when they're there, making them the immune effector cells more powerful, they can overcome all the barriers that block them from functioning. I don't know if that helps, but that's my as succinct as possible summary.
Thank you. So you transitioned to then from working for the NIH, then into roles at Refugee Biotechnologies, then Kite Pharma, and then lately as CSO of Sonata Therapeutics, which is a company focused on cellular microenvironment. So can you describe a bit Sonata Therapeutics technology platform and this focus, which seems kind of highly disruptive around cellular microenvironment, which is kind of back to small molecule in a way?
Yeah, Sonata is not about only small molecules. It's more about understanding how you modulate what we call network medicines or cellular network medicine. How you modulate the cellular network with cancer being central orchestrator, and but all the benign cells around the tumor, kind of in some ways balancing the nurturing the tumor growth versus rejecting it. So, right? How you can you modulate that in favor, of course, of rejection in different ways? Could be small molecule, medium, big molecules, whatever it is. We can talk about that later. But the idea is more how you achieve the 4 Ps, making T cells be there. If they're not there, making them even stronger and overcoming immune suppression if already there. So that's the focus is, and there are different platforms, but that's what we are working on. And the reason why it's so important, and that's actually why I left Kite and Big Pharma. I wanted to go more in a boutique kind of place where you want to focus on the science more than the broader development of therapeutics, and address what are the real problems. And the point I always make is that if you don't achieve the 4 Ps at the same time, you're never going to reject cancer. That's why cancer cure cancer or whatever, have a major long-term benefit of the cancer therapy, because you need to have everything. I always give the example of duck hunting. If you want to get a duck, you need a gun, right? There's no question about that. But just having a gun doesn't mean you're going to get the duck. You need to know how to shoot. The duck must be there, and there should be no fog so you can see the duck. And that's why most therapies don't achieve success, because nobody wants to look at holistically at what you really need to do to achieve that. And so that's what we're working on that. And, I can give more details later on. Some details may require a CDA, somebody interested, but that's what we're working on. Fundamental biology of what needs to be done.
I understand. I'm really kind of interested in understanding more, but I understand that some of the details are confidential. So if we focus on like oncology and immunology, like working across modalities, like small molecules, monoclonal antibodies, and cell therapies, can you see different potentials for different modalities? Like if you compare small molecules, traditional biologics, and cell therapy, would you agree that there is potential for each modalities and perhaps some limitations of each of these modalities?
Yeah, so I look at the problem a little bit different. I look more from the biological standpoint, right? So it's not about, again, a small molecule versus big molecule. So, whatever other, and, protein degraders, whatever comes now, all the buzzwords that are out there. But I think it's more about what we are trying to achieve. So the way I see it more is the breadth of the mechanism of action. So cancer is one thing can do very well is escape around, is mutate very fast. It's a very fast evolutionary process. So if you try to target cancer with BRAF inhibitor, well, it's going to come back because it's going to make the BRAF pathway irrelevant. There's going to be few cells that are going to get around it. Same thing if you try to target the cancer with something, with a MART1 or whatever, some kind of mechanism that target a surface antigen or a MHC-presented antigen, the cancer is going to get rid of that. So having a focused mechanism of action is least likely to work because the cancer is going to get around it. So the reason why then successful therapy works in immuno-oncology in particular, you have these long-term responses because you amplify the direct mechanism. You tackle with one mechanism and then things happen around it. A good example are T cells, right? They go there, they kill the cancer, but that's not why the cancer goes away, because they produce a lot of inflammatory molecule, interferon gamma, TNF, they recruit many more immune effector cells, and the cancer is strangled by too many mechanisms of actions at the same time. It doesn't have a chance, or it has much less chances to survive, to basically escape. So that's what I see, the difference that I see. Now, so going back then to the modalities, I think the how much a modality can achieve this amplification of mechanism of action, the more likely is to be successful. That doesn't mean the small molecule cannot do that. And in fact, actually, one thing that we're interested in Sonata is how can you test the hypothesis you can develop small molecule besides killing the cancer. They also cause what people refer to immunogenic cell death or inflammatory cell death or whatever, some ways that they release those kind of messages similar to what T cells would do otherwise when they get into the tumor, that cause the recruitment of more immune effector mechanisms. So that's, I think, the way I see it. So the more you can have, therefore, a small molecule can achieve this, good. Can a small molecule do that versus if you attack the cancer 20 or 30 different genes against the cancer at the same time? Maybe not, maybe yes. We don't know. That's what we're working on internally. Many other people probably do the same, but that's the way I see it. So that's how I would judge, therefore, what the product is. How far can this product go in amplifying the anti-mechanism? Now, the one thing to keep in mind, however, there are a little trick here. There are, if you look at anti-cancer agent, particularly the biologics, some go for direct cancer killing. So, if you look at T cells, ADCs, or all, they go for the killing of the cancer, and hopefully something comes downstream. And so you can be what I call linear cancer killing or amplification of cancer killing. But then there are other ones that are instead indirect cancer killers, like PD1 blockades, CD47, and blocks the non-immune signal. That's another approach, because those approaches still think to be an amplification. If it works, you have much more activation. The T cells get activated. The more a lot of inflammatory and homeostatic cytokine, other T cells get activated, or other immune effector cells and get activated. The problem with this approach, however, in direct cancer killing, is based on three premises that don't happen all the time. First of all, the target cells have to be there, the benign cancer cells to be there to kill the cancer, right? So that's why PD1 works only rarely, because most of the time T cells are not there. There's no PD1, so it's not going to work, right? Number two, the mechanism of the blocks the T cells should be the preponderant one, the ones that is the most important one, and that's not always the case. So you can have, therefore, tumors that have T cells, PD1, PD-L1. It still doesn't work because there are many other checkpoints at the same time. There are Tregs, myeloid suppressors, cell metabolic inhibitors like ADO, NOS, and so it doesn't work. And number three, the other problem with this is that the idea is that when you target the benign component of the multicellular network, you expect that to be different from the other benign cells counterparts in the rest of the body. That's not always the case. We do have subliminal inflammatory process or infectious problems that actually cause those T cells to look those immune cells to look just the same as the cancer cells. And so that's where we have a lot of side effects. So this is the way I look at the big picture. So are we doing direct cancer killing or indirect? I'm more for doing direct because the only specific things to know about cancer is cancer. And number two, can you amplify? Within their realm, then you judge any product there, whatever they are, and what can they really do. And we do have proprietary way to study internally exactly in a quantitative way what everything is going to be doing with in vitro as well as in vivo models or ex vivo models, if we put it that way.
Thank you. It's great to see kind of a different approach, because I think lately, because there's been sort of a renaissance of small molecules in a way, the people still kind of debate, okay, small molecule, they are cheaper, CMC is not an issue compared to cell therapy. But I think at the end of the day, what matters is treating the patient. So we should kind of look beyond just the modality and comparing just cost and have a holistic view. So thank you for clarifying that.
Let me clarify this. So you're right. So of course, if you can take aspirin, you cure cancer, why would you do adoptive cell therapies, right? So the whole idea. So I'm not dismissing that. It's very, very, very important. Small molecule has also an advantage where their pharmacodynamics are much easier. So it's much more predictable when they do. They are much more consistent in your product than, a cellular product. So there are advantages. The question is more that can we achieve that? That's where we're really working hard. Sonata, and I'm pretty sure somebody else does. We're trying very hard to understand how far can you go with the linear type of approach that whoever amplifies versus, something that comes already like, adoptive cell therapy with T cells. There's a lot of things at the same time ready to start with. But the bottom line, how you balance that, how can you? So I'm not dismissing small molecules as being not likely to succeed. It's just more appreciating, understanding what is the right targets, how can you kill the cancer in a way that's going to be more inflammatory, more immunogenic, and so forth. So yeah, but I'm kind of agnostic. I think you have to try everything from the basic science, and then you develop your product, your modality, when you have something that is going to be a breakthrough.
Yeah, it's like different tools. I agree. I had personally totally agree with that. And from an R&D kind of point of view, I think you touched on the pharmacology, pharmacodynamics of a small molecule being more predictable. But if, like as a CSO of a biotech company, having to develop, or in general, like an R&D process, if you compare an R&D process of a small molecule with the R&D of cell therapy, how are they different? Is it like just a matter of more expensive, or actually there are other differences that perhaps is worth analyzing?
Yeah, well, it's pretty much like you're right. It's completely different. If I have to compare, it's like trying to create an arrow to shoot something, which is small molecule, versus a Ferrari, which is a cell therapy, where you have to have a bunch of other things. So they're so polyfunctional. So, but most importantly, cells obviously are not as controllable as a chemical, is that right? Small or even bigger one, like antibodies. They are kind of known entity with known predictable pharmacodynamics, predictable therapeutic index, because you know exactly what they're looking for. But if you look at immune cells, T cells, anything can change. Even the same cells with the CAR T, with the same CAR, depending on many CARs are there, many antigens on the other side of the balance, everything could be a different story for every single patient, every single cell in the product of a patient. So in reality, yes, adoptive cell therapy is a completely ballgame. It's a different ballgame. And even the potency, as I see one of the questions about that, is not definitely going to be as controllable as you have to have some range of function more than actually what is the potency controllable on a small molecule or on a stable product, because it's going to be different for every patient. And that's where the big challenges are, because it's going to be somebody mentioned reading the questions eventually. You're going to have a lot of differences there in heterogeneity that starts from all the way, but the beginning, from the product from the source of the materials, right? Different patients are going to have different T cells, immunological background. The product is going to be different from the beginning, and that influences a lot what happens in your eventually product development. And then the product development is, no matter how much you try to be consistent, it's going to deliver different kind of final product. And so you work more on ranges of acceptable potency versus real potency. So not only are more expensive, but are more unpredictable. And there are ways to try to get around there, ways to modify the cultural conditions, the way, the timing, and so forth, that you can make it a little bit more predictable. But those are obviously challenges. They're not challenge that you confront when you're making a much more less complicated product. And that's not only about cell therapy. Other what I call complex therapeutics have the same challenge, where you try to achieve many pleiotropic effect of your products, all right? And so so how can you balance that, not only at the level of cost, because obviously very expensive, control of toxicity, and I'll mention what I mean by that. How do you balance that with the benefits of it? And control of toxicity is another subtle issue in adoptive cell therapy. The problem in adoptive cell therapy now is that when the product, T-cell, CAR Ts, TILs, are given to the patient, it's like you put the horse out of the barn. You have no control whatsoever what happens afterwards, all right? Now, there are ways to do better, and that's a paper we just published in Cancer Cell. We describe mechanism to have a control of what happens after the horse is out of the barn. But all these things are normally much more difficult to manage than when you have a product that is stable, like a small molecule, an antibody, where things are going to be exactly what they're supposed to do. And so, there's with something between. And so, yes, it's different. Is it worth it? Probably yes, unless you can find some ways to reach a compromise. And that's also something we're very interested in at Sonata. So basically, is how can you make something that works like a cell product, but in fact is more controllable, is a kind of a imitation of a cell product that does exactly the same a CAR T does, or TIL or whatever, but you have much more controllability but still maintaining that kind of pleiotropism that you expect from the power of the cells. They have a lot of functions, and they're very happy to expand more on the CDA, but I cannot go farther on that.
Yeah. I understand that. So that, it's like a cell therapy without the cell, essentially, like to get to the same mechanism of action. I personally believe that also in terms of biology, as you said, it's more difficult to control what a cell is doing, even the same cell, as opposed to what a small molecule is doing. Probably over time, as we learn more about the biology, we'll be able to control the biology. But it's true that at the end of the day, what matters is the end goal. If I can get it done with a small molecule in a way, do I still kind of really need a cell therapy? Or maybe sometimes I will need both. But there is certainly lots of uncertainty right now in terms of the development, especially at early stage. There is still a lot that we don't know about the biology. And one of the challenges in developing cell therapy that many developers struggle with is developing a reliable potency assay, so a potency assay predictable of clinical outcome. I discussed this topic also with Raquel Ibanez from Stanford. What would be your comment on that? What would be your advice? How can we develop potency assay?
Well, that's a good question. So the potency, well, first of all, potency assay is not necessarily about prediction of response, but it's more about, is the product doing what you expect them to do? Within what range you can comfortable? They say, well, if you give whatever something, it's going to do exactly what you claim it's supposed to do. Is that right? And that's actually what potency assay are now. I mean, it's pretty much you say, okay, well, the T cell, if there is an appropriate antigen, it's going to make interferon gamma. Okay, so I'm not saying it's good enough. The FDA has a lot of thoughts about that, but on the other hand, that's the idea. Is not now. If you have a potency assay is even relevant to the efficacy, that's even better. But you'll get more into some kind of if I don't know, something a little bit more advanced than what the potency is now. How can you do that? Well, the only way to be able to predict the quality of, to look at the characteristic of the product and predict its efficacy is if you have any efficacy, is right? And so the problem is, if it doesn't work, it doesn't matter what it is, still doesn't going to work. And that's what happened in solid tumor now. Most of the time it doesn't work. And if it works once out of 20 times, before you get enough data to really predict what happens, it's going to take time. It's going to take time now, but it works very well for B-cell malignancies, for the CD19 targeting or BCMA targeting products, because if you have enough responses and no responses, you can come up with some prediction, some kind of quality. And so more and more potencies converging characteristics have been published, at least for CD19 from Kite and other groups, that if you have a T cells, CAR T, in the end product that is more on the less differentiated effector status, but more into the stem cell-like status, you're going to have a much more higher likelihood of a not only a response, but also long-term benefit. So a little bit at a time, you can start honing into characteristics. They're just not making interferon gamma if you have the antigen there, but it's what beyond that makes it better cells. But that's not going to improve until we have enough responses to really test them. And this is a very interesting thing, because actually the stemness concept was actually described way before the CAR-Ts on TILs. Well, that was a work we did actually with Nick Restifo and in my lab. We're using the TILs from Steve Rosenberg group and testing what we call the retention samples. So there's some TILs that were given to the patient, some were saved, and we studied them and looking what was the characteristics that could predict in a large number of patients, because TILs at that time were already in melanoma, and through melanoma you had enough cases you can make some conclusions when you studied them. And turned out, surprisingly, we found that the TILs that predicted response in patients were exactly opposite we're thinking of, was the completely immature, naive type T cells. The more TILs that were activated with interferon gamma, even before putting in the patient, the least likely were the patient to respond. I mean, almost a dichotomy. If they were differentiated, the patient would not respond. And that's how the discovery of stemness like T cells started with Nick Restivo's group and Luca Gattinoni. So I think you can hone into some kind of potency assay that is more, but it's only going to happen when you have enough cases so you can actually study it. I think we're getting there for B-cell malignancies or multiple myeloma.
Thank you for that. In terms of collaborations, again, there is sometimes potential to collaborate. Do you see there is a potential to, again, what we were discussing with Raquel, would it make sense to collaborate, and in order to accelerate the transition, to share samples and what would be the conditions to kind of establish collaborations among different centers that could potentially, or for example, share samples and patient samples to accelerate the translation? Would it be possible? Would it be feasible? And what would be the requirements, the conditions in order to make this successful?
So that's such a timely question. It's so fun. It's so interesting, because I don't know if you're familiar with the famous African quote that it takes a village to raise a baby, right? And the interesting thing is they don't talk a city, a town, a country, no, a village. And that's really important, subtle thing. People that know each other, they live and believe and trust each other. And so I think that's what I actually, or some of my always give, it takes a village to grow the perfect CAR-T. So what do I mean by that? It's a lot of effort being done to develop consortia helping each other. And the big stakeholder are academia, or biotechs, and big pharma, is all right? And there is a very, although in theory they can help each other a lot, there are a lot of barriers, IP barriers, legal barriers, and everything. And so things don't really happen as much as they should. And, and unfortunately, that's sad, because the level of expertise that big pharma has in developing product, particularly complex therapeutics like CAR-Ts, cell therapy or other gene therapy with complex delivery systems, they're very good at doing that, but they don't have the, if you want, motivation to be as innovative, to try to new things, right? So how you can combine that? That's very difficult. And one thing that I found as member of the Society of Immunotherapy of Cancer, for a long time, for instance, from the beginning of the society, I've developed many task forces, and I found that those can help. And the trick is not really, is first of all to bring people together. They've never met each other. They're coming from fields that they're very relevant, or whatever. They don't seem to meet at their regular, usual meetings. example was many years ago, cell immunotherapy. It was all about T-cell immunologists. There was no gene geneticist. Although cancer, I keep saying, is a genetic disorder. It's not an immunological disorder. The immunology comes is an epiphenomenal response to the cancer, but actually cancer. So eventually we start bringing people like this, and then germline experts and things to understand more the complexity of everything. So in having a forum where people meet each other and become a village from a city, that's what is going to be the trick. So recently at the annual meeting of Society of Immunotherapy of Cancer, like 2 weeks ago, whatever, it was about 2 weeks ago, we held a workshop together with the Champalimau Institute from Lisbon, Marcos Maurer, where based on the paper we just published in Cancer Cell, how can you raise the perfect CAR T? But that's as an example in general of complex therapeutics. And how can a society like Immunotherapy of Cancer or other ones, of course, if they're interested, try to facilitate the process? And the idea is to bring in people from different area of expertise just to help each other in a pre-competitive way without worrying too much about it. Of course, there are always financial consideration, but in the end, many things are just common sense, sharing common sense, all their experiences, helping each other. And actually people help each other in that case because they start knowing each other, they become a village, they know where to, when somebody calls them they pick up the phone. And so that's, I think, is going to be, and we're trying to develop a task force at the Society of Immunotherapy of Cancer exactly to address this issue.
Well,
Because
thank you for that.
finish, the point is that what I've seen over and over in my career, having worked in academia, with the government, not academia, similar, then in big pharma, small pharma, and biotechs, it is so interesting how disparate their capabilities are. But unfortunately, I found more and more, I've seen so many biotechs fail, not because of the science, but simply because of the resources. They cannot get resources. People wait for the clinical data. It's not going to happen with adoptive therapy. It's impossible to get from a biotech going to a clinical trial. What it takes to do it is very, very, very difficult. And actually would be, big pharma can do it in a snap. So it's not much faster. And they know all the know-hows. So how can we help that process?
Like when it comes to innovation, I think you always need to kind of think outside the box. And I like this idea of consortium, task force within the Society of Immunotherapy.
And, by the way, if anybody's interested, please contact me, Marcos Morer. We haven't started this because we have to go through the process to make sure the society decides to do it through the executive council. But we are proposing this, and hopefully we're gonna get it started. And we did many task force before. They were very successful.
Good. I'm very pleased. So, from the development point of view and translation of science, that's certainly critical. But when it comes to cell therapy, we often discuss also the adoption of these therapies because they are still not first-line therapy, and they are not as widely spread. Right now, like all the approved ones, all the approved CAR T are for solid tumor, for liquid malignancies. You used to be the CSO at Kite Pharma, so you are very familiar, at least with two of those approved. Now we are approaching CAR T, and we are exploring whether CAR T is going to work for solid tumor. Then there are also some approach for autoimmune disease. But in general, my question would be, what do you make of these approaches, and what can we do actually to enable widespread adoption of these therapies? What can we realistically do? What are the key ingredients to make sure this therapy actually going to be massively adopted and widespread?
Well, the basic criteria has to work. If it doesn't work, I think other important consideration to see costs are important. But if it works, trust me, people would use it. The issue is that besides the hematological malignancies, so far hasn't worked, and for many reasons. I think one of the most important is that the T cells don't go to the tumor. And what I used to say, we put the CAR T in front of the horse. If they don't go there, nothing's going to happen. And that's been shown, is a fact. I mean, we see Rosenberg in the early 90s, papers was published where TILs were labeled, tumor-infiltrating lymphocytes were labeled with indium and followed to see where they go. Half of the time they didn't go to the tumor, and zero the patient responded. There's no such thing. So if they don't go, and that's melanoma, if people for which you could raise TIL, successful raised TIL, can you imagine that in the other cases. So, and nobody studies trafficking as well as it should. But then there are the 4 Ps. You have to look at all of that. So until that works, and it's done in a very structured way. So you have to know molecular target of cancer, understand what cancer, understand what sometimes they work and then don't work. We're never going to go very far. So because otherwise we just going to keep trying, shooting in the dark and see hopefully it's going to work something. But I'm confident it's going to work for a very simple reason. We have plenty of evidence that when they work, they work. Number 2, in animal preclinical model system, when the model is relevant, they work perfectly. The only reason why they're working in humans and solid tumor that much is simply because, well, that's not a model system. They have to adapt to understand why different people are different and what are all the issues related with trafficking, penetrance, persistence, predominance, and all that. But I think we can be successful, or you can bypass the adoptive cell therapy by doing something that could mimic the adoptive cell therapy using different approaches that are very feasible. But then here we get into very fancy synthetic biology. Happy to talk about that, but that's going to be another chapter. So I think that's what needs to be done. So we still need to be positive on the concept. The question now is how you do actually get, instead of going to the moon, you get to the Andromeda. How do you do that? Okay. And so that's something that, that is going to take a lot of what we were talking before. Creativity from the academia and biotechnology, and ability to assemble everything from big pharma. That may happen one day, but, there are a lot of hurdles in that direction. Not scientific and practical.
Yeah. It's a bit like when they say about clinical trial. If clinical trials fail, sometimes it's nothing to do with the actual molecule not working. So
why did it fail? Why did it fail? It should not have failed. If you have the mechanism works, it shouldn't fail. There gotta be a reason, but nobody knows. If you ask why didn't fail, nobody knows. Nobody even tested why is the mechanism actually working, and the T cells go there, whatever. So they're never. It's always kind of this. Kind of more trying, a kind of open trial kind of effect approach.
Yeah. And so you said, like, if it's going to work, medical doctors will use it. So there will be a way to kind of fund these therapies, even if they are very expensive. I mean, inevitably costs will go down now that we have more advanced technology to manufacture these therapies. But so it seems that you see the cost component as less of an issue compared to kind of the efficacy of this component of this product.
It's the efficacy. I mean, if you think about, the cost is not that much. If a patient with cancer that takes chemo and keeps recurring and recurring and recurring, in the end, is very, very expensive. It's not true. Maybe a single therapy is cheaper, but if you have the beauty of adoptive cell therapy or these kind of complex therapeutics that can elicit a lot of mechanism of action, in general, when they work, the patients do it very well. Their recurrences are very rare, and nobody wants to use the word cure, but I've seen people that 30 years after having metastatic melanoma everywhere, they're perfectly fine. And that happens. So in the end, maybe it's expensive, one round. And the beauty is also, if it works, it works. If it doesn't work, there's no point doing a second one because it's not going to work. I mean, there's going to be some discussion about that, but pretty much that's my experience. So, I mean, the cost is relative. It's more, does it work or not? I mean, in a way that is...
that's great. So it's more like the traditional, like, cure. If it works, the patient is going to be kind of cured, and it's not going to need any other treatment. which in turn ends up being even less even less expensive than other
Yeah,
treatment
you
together.
can look at B-cell
So
malignancy.
now, no, sorry, I would say now kind of switching gears little bit, going back to where you were a medical student. Over your career, you certainly witnessed lots of breakthrough innovation, and when it comes to cancer treatment and immunology. What do you think were the main drivers of this breakthrough, especially if you look at innovation in oncology and immunology? What made that possible over the past 30 years?
Well, I mean, you're talking about immuno-oncology specifically, is that right? Well, I think,
Yeah, you can focus on immuno-oncology. Yeah.
okay. Well, I think what made it possible is just believing in what is in front of your nose, is that right? The best example, Steve Rosenberg, anecdotes where they saw a patient that had pancreatic cancer before or reoperated 10 years later. There was no cancer. Anybody knows why. I mean, there's something that had happened. He believed in that and worked the rest of his life trying to reproduce that phenomenon. Same thing for me. I went to be a surgery branch. I saw 1 out of 10 people responding amazingly, which is not very common, but still reproducible. And at that time, the skepticism has always been, oh yeah, but it's not good enough. It's not. But yeah, no. But still, you have a biology there. You have believe in you what is from your nose and try to understand what happens, and then you'll make it work. And I remember a quote from one of my original mentors. Talked about just after medical school that he was very famous, and he actually had to face a lot of, if you want, skepticism until he succeeded. And so one of the quotes he also said, he used to tell me is, Franco, sometimes the impossible is just around the corner. So, but people will tell you, will do everything. I have plenty of examples in my career. People are going to tell me, oh, this is never going to work. Until eventually work. And then when it works, say, oh, well, it's obvious. Everybody knew that. Like, what, you just told me few years ago, it's not going to work. So that's a problem that you see is these shallow ways or some way in which people judge facts by reacting and saying something rather than really thinking through every little detail and try to understand what you need to do to make things work when they don't work. Now, if something doesn't work ever, forget about it. But when something works 1 out of 10 times, you better think about and try to understand why. That's what I learned the most.
Sounds like it would
made a lot of progress, I think, in the last, not me, I mean, in general, in people that believe in something and really hone in the understanding what is the mechanism, why things happen or not.
So it sounds like the major driver was down to the motivation of people like yourself and other esteemed researchers that worked and believed that something was going to work, even if it sounded impossible, despite the skepticism of other people. So that was the major driver of, let's say, the major kind of breakthrough in oncology. And now, if you look at, like, in general, in kind of medicine, do you think that like medicine evolved quite quickly over the past 30 years, or like in general, if you look at the field of medicine, let's say everything that we have today was kind of predictable 30 years ago? With technology, it's probably easier to think, okay, maybe what we have now, even just, say just with social media 10 years ago, 20 years ago, it was not predictable or was predictable. When it comes to medicine, what's your opinion? What do you think about the state of medicine today? Could have been predicted something like 35 years ago?
Not really. I think a lot when things went very much faster with the development of new technologies. I mean, the high throughput technologies, way in which you can study diseases and their complexity rather than the classic experimental approach where you look at one gene for all your life and try to try to rationalize that gene explains everything in the world, including global warming and everything. So here is more you do whole genome stuff. That was a major revolution in the late 90s, early 2000, where we were starting to look at the disease as a network. Well, now we call it network medicine. If you want computational biology, ways in which you can get data. First of all, human data, not only experimental data. That's why I started the Journal of Translational Medicine more than 20 years ago. Focus on humans, biology, with technologies that allow you to look at the big picture, the forest and the tree. So that was a major revolution. And this revolution is continuing because it's getting better and better. You can cheaper and faster. You can do single cells instead of, so you can understand what the cell is doing and everything, and how interact with each other in complex environment, like not only cancer, it could be fibrosis, could be many other mechanisms there where the disease is complicated. It's not just one culprit, but the way cells interact with each other and maintain a pathology or eliminate a pathology. And so I think it's been going very, very fast. And if to be honest, I have to say that nowadays is more, the problem is not as much as by the biological level, but it's more at the implementation level. I think we could do much faster progress if it was a much better interaction between the different stakeholders. And so funding, wiser way in which people invest money based on science rather than gossip. I see a lot of, when I listen to investors talking, they repeat like words that I hear around, but they don't even understand the concept to start with. So there is a line that, the failure rates are big, so there's skepticism. So I think the ability of actually understand what you're doing and say, no, we're not going to fail. We're going to make it happen because we understand the science first. I think that's what we need to do because we know so much more now than we knew 10 years ago.
Okay. That's encouraging. Now, of course, in a way, the obvious question is, if we were to imagine oncology and immunology in 20, 30 years from now, what do you think? Like now we are more than predicting, we are just imagining what could happen, what oncology, immunology could look like based on what we have today and where we are going. Where do you see? How do you imagine oncology and immunology in 20 years?
And again, your question. I think we're on the verge of making major progress in curing cancer. I mean, whatever, treating cancer in a biologically meaningful way. And the real secret is going to be, it's not about adoptive cell therapy, small molecule, everything, is cancer-specific delivery. So you can cure, anybody can cure cancer, right? You put atomic bomb on the patient, and cancer is gone, right? But you kill the patient. So the therapeutic index has been the big challenge. And more now, there are ways in which you can become much more specific in delivering. And you can talk about, there are hundreds of papers, even this year, about 300 reviews on this concept come out in PubMed. But how can you be specific so you can bring what we know it's important to get rid of cancer? It's going to make it happen because we know there are some processes that 100% will eliminate cancer, but they are too toxic. You cannot give them to the patient, it's going to kill the patient at the same time. So I think, but when we unlock that in a very precise way, I think we're going to see a lot of progress. Could be ADCs, could be whatever, other people are trying to do. Could be more complex delivery system that are beyond just an antibody or whatever, or a chimera, but it's maybe nanoparticles, something like that. So there are more, of course, adoptive cell therapy. There are different ways. Again, the problem with adoptive cell therapy, major problem is it doesn't go to the tumor. Most of the time, they don't go there. So unless you change the microenvironment, they don't go there, and so that's their limitation. So when you have something that is better, easier biodistribution, you can do that. So I think we're at the verge because a lot of people are working on delivery systems that are specific, and then at that point, you're going to have a much better therapeutic window and possibility to create this direct exponential killing that is going to eliminate the cancer. Use recruiting a lot of mechanism of action only at the cancer side.
So yeah, like highly precise cancer killing. So yeah, let's that's a quite interesting concept. And now, like I guess my very last question would be, in a way, on behalf of all the researchers, all the scientists, medical doctors listening to this fireside chat, and some of them might consider creating a company, like a biotech company. They might be co-founders, CSO. What makes a great CSO? What skills should they master? What they should focus on? And also, I guess every job has its own challenges. So what is challenging for a CSO? So I guess kind of 2 or 3 questions in one.
Well, there are different kinds of CSOs, right? You're talking about biotech, somebody transitioning into developing a biotech, but not a CSO, big Novartis. So in that case, I think what I find, there is a lot of inexperience, of naivete in what it takes to bring a product to the clinic. That's the most common thing I notice. So a lot of these small biotech are what I call a one-trick pony. They think that this great thing is going to be very important, right? And that's great. And it is important. But unfortunately, particularly if you do adoptive therapy, complex therapeutic, forget, maybe it's one more molecule, that's another problem. If you have a target, you can make a small molecule, well, you're going to need a lot of help, and you're not going to do it in your kitchen. You're going to need big pharma to help. Somebody actually has a lot of expertise making a chemical that has the potential to be useful. But if you get into the complicated one, you forget. It's like saying, okay, I have a great way to make a better injection system for a Ferrari. Yeah, but you still have to make a Ferrari, is that right? And that's where a lot of small biotech fail. This naivete, understanding how complicated it is to do everything else. And when they appreciate that, they tend to do it by themselves. So they try to reinvent the wheel. So I find in adoptive cell therapy, I don't know now, but 2 years ago, maybe there are 300 adoptive therapy companies. I guarantee everybody was doing the same, trying to reinvent the wheel, how to grow viruses, how to transduce cells and so forth, which actually is very well known. That's the other thing. Don't be isolated. Don't be a one-trick pony. Appreciate the complexity beyond your, whatever your the mechanism that you're trying to address, but everything else, don't try to invent the wheel. Try to find ways to be humble. Try to get as much help as you can from anybody else. And it's what I used to call, when I was in age, do cafeteria science. Phone calls are cheap now, in fact, are free. Just make as many phone calls as you can, and don't repeat mistakes other people have done. And people actually will help you. Most people will help you. In the non-proprietary way, of course. We're not going to tell you exactly what they're doing.
Yeah.
But in general, tell you, don't do that. We'll be already, and it's not a good idea. You have no idea many times, particularly as a CSO and small biotechs, just making a phone call tells you immediately what you should do or not shouldn't do, because somebody will tell you, nah, that's not a good idea. Or, yeah, that's a good idea sometimes.
Well, I like the cafeteria science. It's a good example, but yeah, it makes perfect sense. Collaboration is, as you said, like within the constraints of like IP and everything. It is always the way to go, and for many reasons, but also not to reinvent the wheel, because and this is usually not appreciated and well this is usually not well received by the investor community, because one of the things investors notice that many companies, they don't really evaluate the competition as well as they should. So they believe that they have the best construct or the best therapies, regardless of the other companies, and maybe they are not that different. So yes, I think it's I really like the comment. So thank you so much, Franco, for these interviews.
Okay, well, thank you. Thanks for me the opportunity. Hopefully it was helpful to somebody, and everybody's very welcome to contact me. And if for more detail, we'll have to go through our BD process, what we're doing, or more in general, contact me for things there. We're very happy to get back to everybody.
And there you have it, an insightful fire chat with Dr. Francesco Marincola, who generously shared 3 decades of wisdom in the development of new medicinal product in oncology. A special thank you to Francesco Marincola for sharing his wealth of knowledge and experience with us today. If you enjoy this content, the best way to thank us is spreading the word about CharaBio and our mission to support first-time founders in biotech. Then follow CharaBio so that you don't miss any of the following episodes. And you're all invited to join us for the inaugural episode of another exciting series, What Pharma Want, featuring the Vice President of Business Development at Merck. This event is free for everyone. Just please register to the link to the video. Thank you, and until next time, bye-bye.